Good evening, everyone, and anyone who's joining us from across the world. Good afternoon, and perhaps good morning to some as well. My name is Udi Wijewardena. I'm the Alumni Engagement Manager for the Faculty of Science. It gives me a great pleasure to welcome our alumni through this webinar series, where the schools within the Faculty of Science have an opportunity to share our up-to-date research with our alumni community. Before we start on the session, I'd like to acknowledge the traditional owners of the land we're presenting on, and also pay respects to elders past and present. And if there's any Indigenous members in the audience, I'd like to acknowledge and welcome you too. Today's session is brought to us by the School of Chemistry, and we've got Professor Louise Bennett and Dr. Alina, who's joining us today to talk about the science of food. In terms of the session today, we've got about 30 minutes of presentation followed by about 20 to 25 minutes of Q&A. So I request you to put any of the questions you have during the presentation in the Q&A function just below your video screen. And when we come to the Q&A section, then we can start off. So don't wait until the end. I'll ask you to put it through, throughout the webinar, so then we can start off with your questions straight away. Okay, well, let me introduce the presenters, just a brief introduction. So Professor Louise Bennett is a professor in food chemistry, and she is the course director for the Master of Food Science, and also the co-director for Monash Food Innovation. Louise's interests of research are physical and functional properties of food, and the specializing space is in proteins and phytonutrients. Prior to joining Monash, Professor Bennett was a senior principal research scientist at the Australian National Research Organisation, CSIRO, working on researching and supporting commercial translation of food innovation. Her research is supported by local and international industry funding, and she's authored about 92 publications and a patent. And she's with an H index of 27. So she's well-renowned in the food space. So we're lucky to have her present today. And alongside her, we've got Dr. Alina, who has been supervised in a PhD by Louise herself. And most of the work that's presented today, some of the work that's presented today has been Alina's research project. So she's actually joining us firsthand to share her experiences with us. So Alina is actually, she's completed a master's in food science from the University of Melbourne. Then she's joined us for the PhD under the supervision of Professor Bennett. And Alina's work's dedicated to exploring the intricate interplay between the nutritional environment and the state of health of an organism. So Alina's doctoral research focused on the development of exome-matched protein blends optimized for maximum digestibility, which I'm sure she's going to explain further throughout the presentation. So I welcome you both, and let's, I'm going to leave the session with Louise, so you can take it over from here.
Thank you, Udi, for that lovely introduction, and it's lovely to be here, and I really do hope everyone finds this interesting. So can everyone see my screen and hear me? Is that going okay? I'll assume so. Okay, so you can see the title of our presentation for today. Now, over the last 20 years, you may have all heard of the concept of feeding your genes. Now, what this has meant over that period of time is the idea of the micronutrients required to keep your DNA honest, to keep it to manage its ability to replicate itself and to produce faithful copies and therefore preserve the ability to replicate an organism. But what we're going to talk about today is the idea of whether our genes can actually inform how we should feed our whole bodies. So how can our genes tell us what proteins that we should eat, which is, of course, a macronutrient supplying our body and our ability to be alive. So I want to explain what's important about protein and why is this information going to help us with the challenges that we face in the future around supplying dietary proteins. And if we are going to understand that concept, we need to understand the quality of proteins and what's relatively good or relatively poor protein quality, and why does that matter? And that brings me to the new tool that we have developed and that Alina worked on in her PhD that we think is going to help the world to use dietary protein in a more efficient way. So what's the problem that we're trying to solve? We need to feed 50 billion people. So I'm sorry, we need to feed 10 billion people by 2050. And what we have, there's a situation where protein is the most limiting nutrient. And the other problem we have is that if the world adopts the patterns of the countries in orange here, i.e. the Western type diets, we're going to need more than the arable land that we have available. We're going to need more than one planet. So this is the urgency that we face in addressing the adequacy of supply of dietary protein. So what is it about the Western diet that confers that particular problem of protein supply? So here's a bit of a snapshot of the environmental impacts of producing different types of proteins. So on the right of the dotted line are the animal-based proteins and the left are the plant-based proteins. Now, quite obviously, for animal-based proteins, we've got to grow the food to feed the animal before we then harvest and eat animal protein ourselves. So these are compounded impacts on the environment, and you can see clearly that for some animal-based proteins, fish, pork and beef, they have particularly high environmental impacts and costs. So we've got these relatively higher agricultural inputs. We've got ethical concerns, of course, about the consumption of animal protein. And then we've got this problem of the growing demand for Western type diets, which include animal protein. And the problem of these Western diets and this animal protein is that it is so fundamentally inefficient. So you can see with the different types here that this is the amount of protein we yield as a percentage of the protein that we feed. So clearly we've got some really terrible inefficiencies considering we've got this challenge of feeding the world protein. So we do need to think and take action now and work out how we're going to solve that problem. So one of the ways that has been is underway to address the high cost of protein of animal-based protein is to start to use more plant-based protein. And it's sort of interesting, I think, that we're trying to formulate and present these in the same texture as meat. But that's just to address, the textures we know and love in meat. So we're now producing these alternate types of protein in various ways. Lots of fermentation-based production, plant-based meats that simulate the sensory properties. We've got all these milks that are derived from plants, and we've got a slew of alternate types of plant, and we're taking the proteins of them. So what I want you to all think about for people online, and Udi mentioned that we do have a chat function. I would be very grateful for you to write down some notes in the chat about what protein quality means to you, this concept, because as we bring all these new kinds of proteins online, I think there's a little bit of a lack of clarity for consumers around the comparative quality of these proteins. So just, put some notes in the chat if you feel like it about what you understand to mean by protein quality, and just do you understand whether consuming high or low protein quality has any consequences for your health? Now, as just to contextualize this, I've got a couple of slides now showing nutritional concepts applied to the other macronutrients. So in comparison with proteins, we've got a pretty good understanding that, that carbohydrates are built of simple sugars. Depending on the numbers of these sugars, we go from a single sugar all the way through to a starch, or different chemical structures, which are the sugar alcohol, right through to fibres. And we've got some ideas about which of these are better to eat or not eat. We have some nutritional guidelines about the quantities in the diet, and we know that we shouldn't really be eating too many simple sugars, those sweet sugars. So we know a little bit about quality of carbohydrates, and some people may even have digestive problems and they become very aware. Now, likewise for lipids, we have a triglyceride substituted with different kinds of free fatty acids. We know about the percentage of fats that should be in our diet. We know that we should eat less saturated fats. We should eat more polyunsaturated fats. So these aspects of protein, of quality of fats, they're also quite well understood and evident in the marketing of foods in the mainstream media. But what is protein quality? So protein quality is an index of how well a protein meets requirements of essential amino acids, as well as physiological needs of the organism, based on an amino acid scoring method, and I'll talk about that soon, comparing essential amino acid contents of proteins with a required pattern that is defined. Mind you, that pattern is only defined for humans and not really for other organisms. We know that foods of animal origin have slightly higher protein quality, but all these scores are adjusted for digestibility. We have adverse effects on quality through storage and processing. And what we do know is the way we manage protein quality is by adding specific amino acids or combining foods to achieve complementarity. But what is the target, and what are the consequences of consuming low and high protein quality? If we achieve the target of a balanced protein of the right quality, we will live and grow with a stabilized body mass. This is as an adult, of course. However, there are consequences for eating either a low or a high protein diet. We move away from that biological balance. So in a low protein context, we can have muscle malnutrition, loss of muscle mass. We can extend our lifespan with a low protein diet, but we have a compromised reproductive function. And the opposite is true for a high protein diet. And not only that, we risk metabolic stress that includes cardiovascular and kidney disease. So there are consequences for not having a balanced protein in the diet. So here are just examples of the types of proteins that we all know and love and eat all the time. Just a food chemistry fun fact is that these animal protein sources tend to be accompanied by fats and water, very low carbohydrate, whereas these plant protein sources, unless they're purified, can come with fats and carbohydrate. So it's a very different nutritional proposition when we move to plant-based proteins. So I've got a couple of slides now to explain protein structural features, so that we can then move into our proposed solutions for understanding protein quality and designing diets around it. Excuse me for a minute. So proteins are assembled from sequences of 20 different amino acids. You can see that there are 9 essential and the others are non-essential or conditionally essential. Excuse me. Many foods are incomplete.
So we have 20 different amino acids in the food, and they are the building blocks of the food proteins. And among 20 of them, there are 9 essential amino acids, which our body cannot make them in the body. So that's why we need to get a certain amount of protein from the food we eat to support the body function. And we also have another 11 non-essential amino acids, and which our body can make them. So this part is not a very big problem to us. And since we cannot make these essentials, and it is very important for us to get an adequate dietary intake of the essentials. And we know that.
I'll try and, oh no, I don't know if I can, excuse me.
So many food proteins, they are incomplete based on the essential amino acid profile, because not all of the proteins, they have a very complete profile that they do not include all of the 9 essential amino acids. And this is especially the case for the food proteins, for the plant-based food proteins, that except for the soy protein, most of the plant-based proteins are incomplete based on their amino acid profiles. And
I'll try and keep going, Alina. I'm sorry, everyone. I'm just recovering from COVID and I've got a bit of a throat problem. So here is a slide explaining protein anatomy. And you can see that the different features of proteins eventually assemble together to explain the quaternary structure of a protein. And these are very important in driving the biological functions of a protein and influencing digestibility and even some food properties. So what we have is a fabulous biological situation where these dietary proteins that we eat are broken down in the body and are then resynthesized into this fabulous set of new and functional proteins. These proteins have to service our blood and the functions of our blood, immune systems, skeletal systems, vision, organs, etc. So think about the biological complexity of this challenge, that this one set of amino acids in our dietary proteins needs to be reorganized into this incredible new set of proteins. So that brings us to the question of protein quality and how do we determine the comparative fitness for purpose of, or the comparative quality of different single proteins and dietary patterns. These are all the methods that are used to characterize dietary proteins. The one that's in use, they've all been superseded except for the so-called DIAS method, digestible indispensable amino acid score. This is an animal-based method involving feeding protein to an animal and then determining or recovering the undigested protein at the end of the ileum. So these are animal-based methods, and depending on the method that is used, the dietary proteins are ranked completely differently. But this is all we've got right now to characterize these dietary proteins. And here's an example of the DIAS method and how different proteins are ranked. So we're comparing a selection of whole proteins versus isolated, measured in various different organisms. So anything above 1 here means that the protein is adequate in terms of all its essential amino acids, whereas anything below 1 is inadequate. So here, for example, in cooked peas, we've only got one of the essential amino acids, and we don't even know which one, is only present at 58% of requirements. So we can see that this is a pretty crude method of ranking proteins and comparing their protein quality. And what are the tools we have right now to adjust our diets to improve protein quality? Well, if we have these animal-based proteins we know are complete, they contain all 9 essential amino acids. But if they are incomplete or don't contain the complete set of amino acids, that is all the proteins with DIAS values less than 1, all we have at our disposal is the ability to mix and match and take a bit of a wild guess to achieve completeness by combining different plant proteins if we prefer a plant-based diet. So where are we at this point in time? We've got animal-based methods for characterizing protein quality. We're not even sure if we want to do it that way. These animal models don't necessarily represent humans. And then what is the essential amino acid target for other organisms? And the real question is DIAS the best we can do? And now I want to bring you into to explain the new approach we've got that is based on bioinformatics and artificial intelligence. So here is an example of the genome and showing us that if we just focus on the segments of the genome that encode for the proteins that are synthesized and made into functional proteins used in the body, we call that the exon subset of the genome. And if we tally up and add together all the amino acids represented by that part of the genome, we can then represent the exome profile of an organism. So this set of amino acids on the left are the essential amino acids, and you can see those ratios, and there are some quite wide error bars in those ratios, but these define a new target for feeding the for the food that we eat to achieve the ability of that organism to synthesize all the proteins it needs. And by the way, there's a reasonable correlation of that DIAS value with the exome profile that we've predicted from bioinformatics, keeping in mind that these DIAS numbers have come from decades of nutritional research. They change all the time, and what we would suggest is that over the next 10 years that these DIAS values will probably come closer to the line and come closer to the exome value. So that brings me to the hypothesis that we wanted to develop and test, is that the optimal balance of dietary amino acids, so food amino acids, are encoded in that organism's exome, and therefore if we match the food to that exome, what kind of biological advantages can we feed the animal and achieve that biological sweet spot, which I explained was the appropriate growth, longevity, and good reproductive function. Now, my colleague, Associate Professor Matt Piper, he's discovered these concepts probably about 10 years ago now. And he's tested them in flies, and he's tested them in mice. But when he did this, he's only used pure and single amino acids. So with my background in food science, and when I came to Monash and learned about this work, it was very clear to me that we wanted to adapt Matt's understanding and turn it into a solution for the diets of people using real food proteins. And just to explain that now that we have the human genome defined, we can ask the question, well, what about the foods that we're eating right now? Are they well matched to our human genome? So if we just look at the blocks of data in the top half of this slide that shows the essential amino acids, where the anything coloured in white or green indicates adequacy, and that we have amino acid levels that are either adequate or exceeding the exome, so that's good. But where we have inadequate quantities, we can see that they're coloured in red. And it's very clear that no single food category and no dietary pattern is naturally giving us that exome balance. So we're not going to get this by accident. We now need to be smart with matching our bioinformatics with some mathematical modelling that's going to let us design diets to achieve that match. And that's what we've done. We have taken the exome of species that we can derive from any species. We've developed a calculator that lets us minimise the difference between the amino acids in the food protein from the exome, and thereby design foods that are feeding the proteins encoded by our genes, and we can do that for any species. So what are the benefits of taking this approach? Obviously, we can feed people, and what I should say is that this ePROB, or Exome-informed Protein Balancing, lets us consume protein at low percentage of the total calories that we consume. And by having low protein diets, we are saving the planet, because we don't have to produce as much of this inefficient protein. So potentially we can feed animals using exome-informed diets. We can optimize ingredient blends. We can save costs for feeding animals. We can apply this technology to food fermentation, And clearly the advantage are potentially less food per organism, and without any trade-off. So we should get the same growth in the animals, the same reproductive health and longevity. And the benefits also are lowering that metabolic waste. So just to recap, before I hand over to Alina to talk about some real data, I wanted to make sure everyone realises protein supply is the limiting nutrient to sustain life. This population growth means that we need to adopt more sustainable ways of producing and consuming food proteins. We need to be addressing protein intake in terms of both quantity and quality. So as I said, I'm very interested in the understanding of the people listening tonight on what you understand about quality, and I guess if this presentation has enhanced your understanding. There are adverse health effects of consuming low quality protein or consuming too much protein. But we now have the solution, which is ePROB, to achieve targets for the diet that are based on this bioinformatics and artificial intelligence. So look, I do apologise for my voice. It's coming good again, but I would now very much like to welcome Dr. Alina Wu, who's going to tell us about the data and research that she did during her PhD. Thank you, Alina.
Thank you, Louise.
And I'll stop sharing.
I will share my screen now. So can you all see my screen? Yeah.
Yes,
So,
we can, Alina.
Good evening, everyone. It's an honor for me to join the webinar tonight and to share the research work that I conducted during my PhD. And Louise has briefly introduced ePROB about its concept, and I will take it over from here and give more details about its application. So let's jump in and see how it works. And here is a little recap for the exome matching technology. And we know that the benefits of the exome matching has been previously identified in the fruit fly in balancing the biological trade-offs between the reproduction and lifespan. The growth benefits were also found in mouse models. And these findings are very promising in the context of human or any other protein-consuming animals, that we can design a balanced protein diet for them just simply based on their genome information. However, the previous exome match diet are designed for the flies and the mice. They are made by the pure amino acid. And in the real world, we are eating the food proteins. So to understand how the real food with blends of macronutrients that can translate into the biological relevant nutrition for us is a major hurdle to overcome. And back to my project, we are trying to work out how to adapt the exome matching principles from the pure amino acids into the food proteins. And that is why we designed the ePROB. There are three major elements involved in ePROB. The first element is the exome-defined requirement for amino acid, and we call it the exome target. The second element is the mixing algorithm. So we have compared the amino acid composition in a number of type of commonly consumed food proteins ingredients, and find that none of them are naturally matched to the exome target. So that means in order to replicate the exome target of an organism, we need to use a blend of different protein ingredients based on their complementary profiles. And that's why we need this mixing algorithm. It is a mathematical optimization tool that can identify the gaps between the amino acid composition in the protein blend and those in the exome target. So the algorithm will try to minimize the gaps and get a closer match to the exome target. The third element we have in ePROB is also the core of the ePROB. It's the digestibility adjustment of the protein ingredients that we want to include in the final blend. The digestibility adjustment is a very big challenge we are facing when mixing the food proteins. This is because unlike the single amino acid, which can be directly absorbed into the portal circulation, different types of the food proteins, they vary in the ability to provide the digestible amino acid that can be used to support the metabolic response, such as the body growth or immune functions. So if we use this total amino acid profile of the food proteins to match with the exome target, the efficacy of the exome matching can be compromised. So in order to solve this, we have developed a method for the digestibility adjustment of food proteins, and we mainly focus on the monogastric animals. In a short summary, it is done based on a prediction of the bioavailable amino acid in the blended ingredients, and it is achieved through a series of the in vitro experimentation that mimics the protein hydrolysis in the monogastric animals. And after the digestibility corrections, we can obtain the in vitro predicted bioavailable amino acid profile of the blended ingredients and use this profile for matching. And in the end, what we want from ePROB is that once we tell the mixing algorithm the blended ingredient we want to use and their amino acid profile, and they can tell us the blending proportion of each ingredient so we can make an exome match protein blend. And we have tested the efficacy of the ePROB formulated protein blend in the laboratory mice. And here shows the diet design. 3 different types of the food proteins are blended at different proportions to generate diets that are matched or mismatched to the exome target. And here we have 2 matched diets in which one of them, the M-adjusted diet, it is adjusted for digestibility. And we also have another 2 mismatched diets. So the way to assess the degree of matching is through a value called the degree of limitation, the R value here. You can see that in the bottom. And it is the value to help us to compare the supplied amino acid to the exome demand. It can also predict the most restricting essential amino acid in the food that can restrict the growth of the animal. So a food with a higher degree of matching means it is a better match than the other to the exome target than the other one here. And as we can see from the graph here, our 2 matched diet have a higher degree of limitation and than the other 2 mismatched diets. And among them, the M-adjusted diet is the best match, that it has the highest R value, means that about 76% of the proteins are predicted as bioavailable. And the figure here on the right shows the diet characteristics. All of our 4 experimental diets have the identical energy density, and they were processed in the same way, and no heat treatment is applied, so we can minimize the negative processing impact. The most important feature of the diet is that the energy proportion from the protein, carb, and carbohydrate are constant in each diet. So in this case, we can assure that for every gram of the food that we give to the animal will provide the same amount of protein energy. And the only difference is that the amino acid ratios vary across the diet. I also want to emphasize here that all of the 4 diets, they are low in protein energy. They are low protein diets that the protein only contribute to a total of 6% in the total energy. Then let's move to the experimental setup of the feeding study. We used the mouse as the animal model, and they received at 2 weeks of age. And after 1 week, we let them to get familiar with the environment. They were randomly assigned to one of the 4 experimental diets. In each diet, we have 6 animals replicates per gender per diet. So the feeding took for 13 weeks. All animals are fed ad libitum, so they have a free access to the food and water and eat as much as they want. And during the feeding period of time, and we measure the weekly food and water intake for each treatment. And we recorded their weekly body weight for individual animals, and also measured their body compositions in terms of the lean mass and the body fat in every 2 weeks. And here shows the result we got, and I will only focus on the feed conversion efficiency here today. The feed conversion efficiency is the ratio of the body weight change to the total food intake from the beginning to the end of the feeding. It is a very simple measurement to determine the relative ability of the animal to turn the feed nutrients in the desired output. In this case, is the body weight. So the 2 figures here shows the feed conversion efficiency for the male and the female animal. And we can see that in the male animal, our M-adjusted, which is the exome-matched digestibility corrected diet, it produced a 62% increase over the worst performed mismatched diet, the blue one here. And such increasement for the female animal reached 40% over the worst diet. So the M-adjusted diet is the best performed diet in both gender. We can also see that when the digestibility correction was not applied, as shown in the diet M, the Exome-matched diet, the black one, it cannot improve the feed conversion efficiency compared with the two mismatched diets. So that means the digestibility adjustment is really a very important part when we want to apply exome matching on the food proteins. So from our feeding study, we can really see that efficacy of the exome matched digestibility adjusted, the M-adjusted diet in supporting the body growth. But how do we know whether this diet is better than the commercial standard diet? So to answer this question, we used a diet called the lab diet as the example to make the comparison. It is produced by the US company and used as the standard lab diet for the mice by the Jackson Laboratory, the largest lab animal distributor in the world. So the table in the middle here shows the difference in some of the nutrition parameters between the lab diet and the ePROB formulated M-adjusted diet. The important message here is that the lab diet is not exome matched, and it has a very low degree of matching compared with the M-adjusted diet. Well, both of them are limited in histidine as predicted by the exome matching technology. And also the protein energy in the lab diet is 22%, way more higher than our diet, which only contains 6% of the protein energy. So considering the huge variation, it will be very interesting to see whether the animal will grow differently when fed these two diets. And here shows the comparison of the body growth. The trajectory of the body weight change of the male and female animals was shown on the left. The red one is our ePROB formulated diet, while the black dashed line is the animals fed on the commercial lab diet. So we can see that the trend in the body growth is similar in the male animal on the top, but it is quite obvious that the female animals on the ePROB diet, they outgrow those on the commercial lab diet. So considering that the initial body weight of all of these body weights are based on the same genetic strain of the mice. So considering the initial body weight of the animals at the very starting point is different, so we cannot use their absolute weight to make a direct comparison. So we further compute the body growth rate for those two diets. This value can tell us the amount of body weight animals put on for each week. So as shown in the bar chart here, we can see that the female animal in the ePROB diet, the red one, they grow faster than the commercial lab diet. So although for the male animals, the body growth rate is slightly lower than the lab diet, at least this difference is not huge, especially considering that the ePROB diet contains very low protein energy than the lab diet. So based on the preliminary results I just showed, I hope you all have an idea about ePROB and its benefit in supporting the body growth. And here are some potential applications of ePROB we proposed. Firstly, it can be used as a very simple tool to evaluate the quality of the protein quality of the current available diet. Also, it can be used to design the feed for monogastric animals because our digestibility corrections is designed based on the protease in the monogastric animals, such as the chicken and the pig. And it is especially applicable when designing the low protein content diet for the animals, since the protein quality is optimized. So here is the brief conclusion. We think it is time to let the industry know that we have a better solution to formulate the animal diet. The design of the ePROB tool is a very good candidate to formulate low protein diet. We know that the feed cost is the biggest expense for the farmers. So when by lowering the protein level in the diet, we can help to reduce the feed cost, and this will bring significant economic benefits to the industry. Also, as we can see from the mice study, when we feed the animals with the exome-matched diet at a very low protein level, like 6%, we can expect a constant or comparable body growth compared with those fed a higher protein, like 22% in the commercial lab diet. So the last benefit of ePROB is that once we lower the protein content in the feed, we can help to lower the nitrogenous content in the feces, and this will automatically lower the environmental burden, like the nitrogen output, and we can help to protect the environment. So I have finished the science part of ePROB here, and this here shows the ePROB team under the lead of Louise. And we have Matt as the biological scientist, and he is the first person who established and tested the exome matching theory in 2017. And we have Andreas from the School of Mathematical Science, who wrote a very beautiful algorithm in the ePROB, so we can make this happen. And we also have Dr. Minoli, who works as a research associate in this team, and myself as the application scientist. And we try to achieve the commercial application of ePROB to different agriculture animal models. And here brings to the end of my presentation, and thank you very much for listening. I hope you find something interesting and from our presentations. Please do not hesitate to reach out if you want to get more information about ePROB, and you can find the emails below. And thanks again. So from now, Professor Louise, Bennett, and I will take any questions from the audience. Thank you so much. So yeah, I have, so shall we start the questions from the audience?
Yes. So we've got a number of questions that have come through. So I'm trying to see at the start what questions we've got. So there's a question about the quality of protein. So just looking at what are some examples of good quality proteins and poor quality proteins? And when you say good quality proteins, what did that mean? So let's start at the beginning and then we'll work our way down through the questions. So Alina, would you like to answer, or Louise, one of the two?
I'll have a go at that one then, Udi, and I'll certainly let Alina answer some too. Yeah. So you know, good and bad or poor quality proteins sort of very simply fall into the two categories of either animal-based versus plant-based. There's an exception in plant-based proteins that soy is a very nice and high-quality protein, but in general, the plant-based proteins, except for the legumes, tend to be incomplete. So this is the challenge we have, that the animal proteins are naturally have higher quality, but we've got to be sure we're not eating too much of them, but and exceeding and creating that biological waste. But in the plant category, it's the legumes that are the highest, have the highest abundances of essential amino acids.
That's great. Thank you, Louise. Now, this question, perhaps it's for Alina. It says, is the exome matched diet something that could be personalized, or is it sufficient to use the human's reference genome?
So the genetic information is quite conserved, and it is within the same species. We didn't, like, at least Matt didn't see a very big difference between individuals, and also the age. Many people ask about whether the changing of the age will influence the exome target, and the answer is no. And the same as the gender, because Matt's research on the flies, he predicted the exome target for both male and female fruit flies, and he has tested that in fruit fly experiments, that they didn't observe any significant enhancement in the growth or the reproductive functions when they do the exome matching based on the male and female respective specific genome. So we can see that the exome target is really conserved. So for within the species.
Right. Okay. This may be a question for both of you. So this is thinking about you know, will improving feed conversion to body weight efficiency lead to more obesity? So that's a more practical sort of question. So
to answer this question, at first, so we want to, that's why we design our diet in a very low protein content. And also from the result, we can see that even so for both the male and female animals, they grow, their feed conversion efficiency is improved and their final body weight is improved. But because of the limit of time, I didn't show the body composition of the male and female animals. So from that result, we saw that the exome-matched digestively corrected diets drives both higher the lean and fat mass in the male animals, while it drives higher body fat and produce a comparable lean mass in the female animals when compared with the worst matched diet. So we think it is highly linked to the physiological difference of the different sex. And our result at least showed that we also analyzed the liver fat in different animals, and we find that our mice is not obese, and also the fat level, as long as you control the fat energy contribution in the diet, and I think the ePROB formulated diet won't lead to the obesity. And so our results also suggest that once the lean mass in the body is locked down, the development of the fat here is very meaningful in stabilizing the body weight and maintain a balanced metabolism in a low protein feeding scenario.
Okay, very interesting, isn't it? Gosh. I've got another question. It may be one for Louise. How does fermentation apply to this method? Like what you mentioned, Louise, given it is focused on monogastric animals, how does the fermentation bit apply?
Right. So I did mention that we might be able to design use ePROB as in principle to design the feeds for a fermenting organism. So in that case, we we identify the organism, we obtain its genome and its exome, and we can then design, the fermentation broth to feed that organism. Of course, in some fermentation broths, they use free amino acids, so we don't. And as Alina explained, the ePROB concept involves this digestibility correction to compensate for the fact that we're feeding whole proteins. So in a fermentation system, if you were just feeding single amino acids, we go back to Matt's work right at the beginning. But if we do start to feed whole proteins, it's a little bit different, because that's correct. It's not a monogastric system, and the enzymatic corrections would be a little bit different. So we've yet to explore that application in fermentation.
Okay, very good. The next question I've got, it says, can this approach be applied to diets of people with different body compositions, such as performance or strength athletes? So it could be a question for both of you, I would say. Maybe I'll let Louise start, and then Alina could probably chime in.
Yeah, look, as Alina said, and I think there was a selection of questions on similar themes asking about that personalized variation, and so would an athlete benefit more than an older person, and so on. The main scenario for application of ePROB for humans is in the situation of restricted nutrition. So if an athlete was, for whatever reason, was trying to restrict their dietary intake, the quality of the protein and ePROB matched protein would be very important. And likewise for older people who have, limited appetite, maybe for, astronauts, people on the moon, there's not enough food, other sorts of situations of rationed foods. That's when there is an advantage in that ePROB type of feed. So, but we wouldn't say, because an athlete's got the same genome, we wouldn't say that it would. We wouldn't be able to design a specific exome for the athlete. No.
Yeah,
right. Okay, good. And that's that's definitely something that comes to our mind when we think about diets that's being planned, etc. I think there's a couple of questions about the experiments itself. There's one question where it's asking how many animal species were used as samples of animal protein. This include insects. The other one is, why do they actually start at different body weights while we were going through the results?
So for the first one, we in the ePROB formulated diet, we only use one type of the mice, which is one of the common genome strain of my laboratory mice that commonly used in the nutritional research. And for Matt's project, he using the pure single amino acid, he has identified the efficacy of the exome matching in mice and also identified in the fruit flies. So as far as we know, it has only been tested in the fruit fly and mice.
Right.
So the food protein of the ePROB design one is only tested in mice. And for the second question, that why the animals start at a different body weight. So I think you refer to the animal growth that I included in the very end, that the comparison between the lab diet and our diet. So in our diet, all of the animals, their starting weight at week 3 is the same, like in the ePROB formulated diet. But when we compare that with the lab diet, this is the published data by the Jackson Laboratory. It is the average data weight across nearly 300 mice. So I guess because of the large number of variation that we don't know what's the condition of their animals. So that's why that I mean we cannot use their absolute body weight to make a comparison. But at least their genetic strain of these two animals are the same. So we can make a comparison on their body growth rate instead of the absolute body weight.
And that's why Alina computed the growth rate instead of the absolute change in body weight.
Yes.
Right. Okay, that's quite interesting. Quick question. This one will be the one before the last. There's one last question at the end. Are plant-based meats good at matching the human genome-defined needs for proteins?
Yeah, no, so as I showed in that summary of data where we compared the exome matching characteristics of different food groups and different dietary patterns, the problem is that the plant-based milks absolutely do not match the human genome. They're much worse than the animal-based milks. So and this was my point, and I'm really grateful for the people who put in some responses about their understanding of protein quality, because I think it's fair to say that the industry doesn't really want to draw attention to the fact that these so-called plant-based milks that are sort of weak extracts of protein from the plants are much lower in protein quality compared with the animal milks.
Okay, wow. I'm sure we all have a bit of an almond latte and all the plant-based milks. So that's something to think about, food for thought. The last question I'll ask both of you is, will ePROB be immediately economically viable, or will it pay itself off over time? So it's more the economics of it.
Well, maybe I'll start, and Alina can add to my answer. But look, we are very hopeful to commercialise this concept and apply it for livestock, for designing feeds for livestock. And it should pay for itself very quickly, because this ePROB technology permits low protein feeding of animals. So what I've learned recently, for example, with chicken feeding, the excess nitrogen in the feed causes burns in the animal's legs and feet. And that's one of the ways we can improve animal welfare. So the short answer is that this is absolutely economically viable, and we can design feeds right now that will that can be prepared and fed to animals, and we do need to do the research to prove the feed conversion efficiencies and efficacies of these new feeds.
Right.
Alina, is there anything you'd like to add to that?
So actually, we have analyzed the exome matching, the degree of exome matching of many current different chicken diet, and we find that none of them is exome matched to the chicken diet. So there is a bit of room for us to improve and use the ePROB to solve the problem, I guess.
Yeah,
that's right. We're almost at time. There's one last question very quickly. Are there any good resources to go to learn about plant-based combinations to create a complete protein? Just very briefly.
Gee, I'm not sure if there are. Alina, are you aware of that?
So I quite confused about the question. Is it to what about the I couldn't see the question here.
It's about, which could you design, which plants to pair together to achieve, to lower the limiting amino acids. So I think that the short answer is probably no, and it again reflects my understanding that there are many, shopper, assistant apps that you've got that tell you about quality of your fats and your carbohydrates, but they don't tell you that exactly that question about proteins. How could you pair up different proteins to achieve, a better overall protein quality? And so, I'm really grateful to the listeners tonight who have helped me and sort of confirmed my suspicion that the understanding and for this, learned group of people is probably the best in the world, but it's still very limited. And I think it's because the industry isn't really helping us to understand.
Interesting. I think the last question, there's another one which is asking about if we published a paper on ePROB. I think the short answer is yes. Sorry, Alina, go ahead. Yes.
We published a little paper on the Food Australia. That is the media press publication, but we are now submitting the manuscript to a scientific journal, and it's in progress.
I'd like to thank both Professor Louise Bennett and Dr. Alina Wu for taking the time in this evening to connect and share your knowledge about the ePROB and the new research in that food chemistry space. So I thank you both. So I hope that our alumni community gathered some new intel and information on this exome-based diet and protein matching. And I hope you can take that knowledge and share with your colleagues and also people at your workplaces, etc. With our alumni, I want to actually share with you a couple of things just before we go. I'd encourage all of you to sort of join our alumni LinkedIn group. So that's where most of the time the events and activities and anything that's coming up, maybe alumni benefits are advertised regularly. So I'd encourage you to join in and invite all of you. And sometimes, I mean, it's a group. Our alumni can actually post anything that of interest for the science community as well. And also if there's any roles or any employment information that, if you're hiring for a role, feel free to actually advertise it in there, because it's all for science alumni. And as Monash alumni, you'll have the tools and the knowledge to actually do a great job in your workplaces. So that's one thing. The other thing I'd like to share is that we will continue to run this webinar series next year. So I'd very much like for all of you to tune in and also provide me with the feedback so that I can continue to improve our engagement programs so that it is more relevant to our alumni. It is for you, so hopefully the feedback will help me sort of continue to improve them so that it gets even more beneficial. I'll share the link to the alumni LinkedIn group. I hope you all join, and I hope you have a wonderful evening, and marks the end of the session. Thank you all.
Thank
Have
you.
a good night.
Thank you, everyone.
Bye.